Hey guys, so today we're going to start on chapter 17. It's the last chapter in this of the semester, in this unit and of the semester. And chapter 17 is actually kind of cool too. So hopefully you guys will enjoy it, right? Because it's all about our recombinant DNA technology, or basically genetic engineering, and how we manipulate genes, basically splice them together, take them apart, right? Isolate them, put them in different organisms. We just do a myriad of things that are absolutely mind-boggling to the mind, at least to my mind. And it's incredible to witness, right? It's incredible to see this new era of genetics just kind of coming about. So genetic engineering is essentially just, and DNA technology is just, again, the technology that we have in manipulating and working with and analyzing the DNA. It includes sequencing, it includes DNA recombination, which is basically putting different DNA sequences together and making one full, continuous DNA molecule. Everything can basically be summed up by the word biotechnology, which is the manipulation of organisms or their components to make useful products. So not only can we actually do research by DNA manipulation, etc., with these DNA technologies, but we can also make specific things like specific drugs, right? We can help make other express proteins that maybe humans need in excess, for example, like insulin. We could also make different actual useful products. One thing that they were working on as well was protein silk fibers from spiders. So they noticed that spider fibers were really resilient, really great. They're incredibly flexible and just elastic and strong. But alone, they just wouldn't when they singled out specific fibers, they wouldn't really accomplish much for human benefit. So they actually spliced two different types of the two different genes that encode two different silk fibers together, and they got this really resilient, really good type of silk fiber. So the applications of biotechnology are basically far and wide. There are so many different kinds of applications that we can have, all the way from agriculture to criminal law to medicine, right? There's so many different things, to industrial stuff. It's absolutely incredible. So you can see here kind of how the field of biotechnology can be split. We have things for forensics, food, environment, biofuel, medical, labs, pharmaceutical, regulatory, right? They're trying to, NASA's trying to change bacterial chromosomes and everything, and transform bacteria so that they can help humans go to space. It's just a lot of stuff. So what is recombinant DNA technology? This is when recombinant DNA is just DNA created by joining DNA pieces from different sources. So one DNA from this source and one DNA from another source, and we put them together, and now all of a sudden we have something completely different. And so this is the technology, like recombinant DNA technology, is the technology that we have to actually carry out and get recombinant DNA. And it's led to major advances in research in molecular biology and genetics. In fact, without really recombinant DNA technology, a lot of the different discoveries that we've had today wouldn't have been discovered. It's extremely important for research processes. The method are used to copy or clone DNA, right? This is imperative because, as you guys know, there's a lot of DNA in an organism, right? There's a lot of DNA in an organism. And just in one of our cells, we have 3 billion base pairs, 3 billion base pairs. And of those 3 billion, maybe you're just looking at one small little target gene that's about 1,000 nucleotides long. So it's kind of difficult to analyze your target gene with all the rest of the genome getting in the way, right? Very difficult to analyze. So what you want to do in order to analyze it is specifically target it and clone only that section, take it away from the rest of the genome, right? The rest of that extra jargon. And then this way we were able to actually have enough quantity of DNA to analyze it and to manipulate it. We can now take our DNA and do something with it. Anyways, so clones are just recovered copies of recombinant DNA molecules. They have allowed scientists to isolate and study specific DNA sequences from a genome. It's used to study structure and orientation of DNA, is used to isolate, replicate, and analyze cells. So in order for DNA recombination to actually work, two things are absolutely critical. We need restriction enzymes and we need cloning vectors, right? So let's look at what these are individually. Restriction enzymes. These are, they started out, they were discovered a while ago. They're basically one of the defense mechanisms that bacteria have against bacteriophage attacks. We know bacteriophages very well, right? We have these viruses that infect bacteria and they inject their genome inside the bacteria. And then as soon as that genome hits the bacteria cytoplasm, the virus just takes control of the bacteria. It degrades the bacterial DNA, forces the bacteria to make more of its viral DNA and more of its viral particles. And then usually if it undergoes a lytic cycle, the bacteria explodes and releases, like the bacteria itself dies and it releases all these other viral progenies, right? So one of the ways that bacteria have actually defended themselves against these viral genome attacks is by making these kind of molecular scissors. So they have these enzymes called restriction enzymes that cut the DNA, the genome of viruses once they come in. Okay, these restriction enzymes don't work against the bacterial chromosome itself, right? They have very specific sequences that are sequences that they know are found on bacteriophage genomes. And so they're DNA cutting enzymes. So they're basically their defense mechanism. And we were able to isolate these DNA cutting enzymes and use them to our advantage because, again, they're very specific. Just like any enzyme, each restriction enzyme is specific in itself, and it will only ever cut a specific unique type of recognition site or restriction site. So that place the sequence of DNA that they specifically cut, each restriction enzyme specifically cuts, is known as a restriction site. And it's actually really fun. These restriction sites actually tend to be palindromic, meaning if you look at them from, there you go. Palindromes is just a symmetry exhibited by recognition sequences. So the nucleotide sequence reads the same on both sides. So we have GAATTC, and then on the other side, GAA going the other way, TTC. So the recognition sequences each is specific to each restriction enzyme, and it's palindromic. And the way they actually even cleave and cut the recognition sequence or restriction site is going to be specific for that enzyme. So for example, EcoR1, which as the name suggests was isolated from E. coli, EcoR1 cuts in a very jagged manner. So it cuts right here at the G, right? And then it's between the G and the A, and then again right here between the A and the G. So it cuts the phosphodiester bond. So it cuts the sugar phosphate backbone. It doesn't actually change the integrity of the nucleotides at all. It just is a nice clear cut. So our cut is nice and jagged, which leaves occasionally, depending on the restriction enzymes, sometimes a 3' overhang, right? So we have the single strand of DNA just kind of like jutting out, all nice and sticky, because DNA doesn't like to be single stranded. It's going to want to find its complementary DNA so it can form double strands. So we call these, sometimes when the restriction enzyme cuts in a jagged manner like this, we call these sequences or these cuts, sticky ends, right? Hint 3, which comes from Haemophilus influenza strain D, also cuts in a jagged manner. You can see that its own sequence is very different from the EcoR1 sequence. And again, that's the only sequence that it's going to recognize. And it cuts again in a jagged manner to give us sticky ends. There are a couple like Hay3 or Hinsi or ALU1, that cut and form blunt ends. And so these blunt ends are just two double-stranded molecules. There's no 3' overhang. These are still useful to us, but they're a lot harder to use in recombination technology because, as you can imagine, the sticky ends, this name suggests, are sticky, and so they want to bind to something else. So what we can do is we can cut a target DNA with the one part of one restriction enzyme, and it's going to get the sticky end. And then we can cut another DNA element with the same restriction enzymes. So these two DNA elements now that you have the same type, like the same ends, right? They have the same sticky ends. So when you put them together in a little test tube, they're going to anneal. And annealing is just joining together. right? So they will just join together, and then DNA ligase goes ahead and fills in that phosphodiester bond between the missing phosphodiester bond between the adjacent nucleotides. Okay, so these restriction sites and restriction enzymes are present randomly in the genome of organisms. They do show up pretty often, and the fragment site produced varies based on the number and the location of the sites throughout a DNA sample. This is also known as restriction enzyme digest, because they're digesting, they're cleaving, they're cutting the DNA. So it's also known as digestion. Okay, so yeah, here you have the definition of sticky ends when you had fragments produced with overhangs, blunt ends, fragments produced with double-stranded ends. And again, these ones are harder to use than the sticky ones. The sticky ones, they like to anneal. They have to anneal with another like complementary single strand, and so you can join things together. Oh, as you can see in this example, so you have this green DNA and this purple DNA, right? From two different species or something. You cut it with EcoR1. You cut this one as well with EcoR1, and they generate ends that are going to be complementary to each other because they have the same sticky ends, and you can basically put them together. And then you can see that there's this missing gap. That's the missing phosphodiester bond. So you add DNA ligase to basically seal the deal. Okay, and it kind of it joins the actual restriction fragments covalently. And then we have intact DNA molecules. So this is the first thing that we needed in order for recombinant DNA technology to work. The next thing that we needed was cloning vectors. These cloning vectors are just DNA sequences. The first ones discovered were basically plasmids that carry DNA molecules. So we can open these DNA sequences and put in our target DNA, and it can be stably maintained within this cloning vector. The cloning vector wouldn't reject it or anything weird like that. It just stably stays. So it accepts DNA fragments, and then in addition, cloning vector, right? So it's not just a vector that holds the DNA. It goes ahead and clones the DNA. There's all sorts of different kinds, which we'll get into in just a second. But the main difference, like the main features of all of the different kinds of cloning vectors, like for example, plasmid vectors or cosmids or artificial chromosomes, right? These are all different kinds of cloning vectors. The main thing that they all have is that they're able to replicate independently. So all of them can replicate independently. Some of them replicate more often than others, depending on the actual cloning vector itself. They have a lot of restriction enzyme sites. So they literally have an area on their cloning vector that is just full of little restriction sites that we can choose and cut. So we can cut it open and then put in our DNA fragment. This is normally called a multi-cloning linker, a multi-cloning sequence, or a polylinker site. And they also carry a selectable gene marker. This helps distinguish host cells that have taken the cloning vector up and those that haven't. So don't worry if you don't understand any of these three different features right now. We're going to go in depth with them as soon as we talk about bacterial plasmids. So bacterial plasmid vectors. Plasmids basically are used to clone DNA. We know that plasmids occur naturally again, right? So this is basically biotechnology. When you look at something that occurs naturally and then you just take advantage of it and for human gain. So plasmids are one of those things that we took advantage of. Remember, they were extra chromosomal autonomous DNA molecules, right? That are outside of the normal bacterial chromosome. They're outside the normal bacterial chromosome, and they replicate by themselves. They hold their own genes, and of course, they contribute to the bacteria's phenotype. So what we did is we could genetically modify these bacterial plasmids, and when we did, we created the first vectors, and we made some beautiful vectors with, that had like the different selectable markers and the multi-cloning site, like PUC18, which is a very common type of plasmid that is used. Okay, so we have a wide range of them with very useful features. They're very readily available. They're engineered to contain a number of convenient restriction sites. So again, we put these restriction sites, a multi-cloning site in them, and a selectable marker gene to see if there's absence or presence of the bacterial plasmid in the host. Okay, so I'm going to change the video now and talk a little bit about plasmids. Okay, so And then I made lots of different colors as well. So plasmids are circular extrachromosomal DNA, right? And in addition, these extrachromosomal plasmid DNAs, they all have a specific site of like replication origin known as the ori, right? Every single one, every single plasmid comes with them, with an origin of replication. Yeah, someone came to say hi. He just showed up.